Hello and good evening to all of you. In this presentation, we will just going to see about the complement system. So the question arises, what is a complement system? Complement refers to a group of a large thermolabile enzymatic proteins which are found in a serum and the body fluids, and it involves an antigen antibody reaction. It also involves lysis and the phagocytosis. It is represented as C. The complement system is a part of our immune system. Whenever any antigen or any foreign particle comes to the body, as immune system works, the complement system play a very important role in our immune system. It enhances the ability of the antibodies and the phagocytic cells to clear microbes and damaged cells from an organism. It also promotes inflammation and it attacks the pathogen's plasma membrane. Uh, Here are the components of the complement system. The complement components constitute 5% of the serum globulin fraction. Most of them are circulate in our body, but they are in their inactive stage, and that inactive stage are termed it as the proenzymes or zymogynes. That means when all the components are present in our body, it is in an inactive stage, which is termed as proenzymes. The complement reactions start with an enzyme cascade. It involves the following factors such as a C1 to C9, and it is designated as some numerals such as C1 and C9. And there are some letter symbols as well. They are the factors D, or they are the trivial names that are the homologous restriction factors. These peptide fragments form by activation of a complement, and they are denoted by the small letters. Here are some of the functions of the complement system. Complement system play a very important role for the lysis of the cells, bacteria and viruses. It also play a very important role in opsonization. It is a process which promotes the phagocytosis of a particular antigen binding to a specific complement receptor on a cells of the immune system for triggering specific cell or for inflammation and for secretion of immunoregulatory molecules. Not only that, but it also play a very important role in immune clearance. That means to remove immune complex from the circulation which are deposited in the liver cells. Now the three major pathways of the complement in activation, which are as follows. The following pathways are classical pathways, alternative pathway, or lectin or MBL pathway. This pathway requires a multiple steps which are as follows. It requires recognition. All the complement needs some recognized site for binding it, some enzyme activation and biological activation. Here we have an image. We can see how the classical pathway works. So it begins with the antibody binding to a cell surface and ends with the lysis of the cell. The C1 have a three binding site, that is C1q, C1r and C1s. It binds to one antigen antibody complex and it activates C1, which further activates to C4, and it leads to the activation of C2, and it further activates C3 and it leads to the C5 activation. So this protein in this pathway, which are termed as C1 to C9 pathway, they are discovered not in the order of the reaction. When the complement is activated, it's split into the two part, the A part, which is a smaller part, and the other part, that is B part, which is a larger part. And it usually the active part. B part is usually the active part. And the three steps are required for this pathway. They are the activation, amplification and the attack. Now here we have another pathway, that is alternative pathway. Alternative pathway start with the C3 complex and it ends to the C5 complex. So this pathway is activated by the bacterial cell surfaces and their components such as endotoxin, microbial polysaccharides and other factors. This pathways can be activated before the establishment of any immune response to the infecting bacteria. The initial activation for this alternate pathway is mediated by a properdine factor B binding to C3b, and then with properdine factor D. So factor B and factor D plays a very major role in the alternative pathway. This C3b sticks to the cell surface and anchors the complex. Then inactive Ba, that is capital B small a, splits into the complex and it leads to the cleavage of activation of many C3 molecules. The complement cascade continues in a manner analog to the classical pathway. The next pathway is all about lectin pathway. So major function of the lectin pathway is similar to, we can say, to the classical pathway. But a major component which play a very important role in lectin pathway is the MBL, which is and the MASP. It forms the MBL MASP complex and it leads to the activation of C3 and it further leads to the activation of C5. The C3 convertase and C5 convertase, this both are the enzyme which play a very important role for the conversion of C3 and C5. The active complex formed by the association which causes a cleavage and activation of C4 and C2. The MASP1 and MASP2 proteins which also play a very important role. And while activating the C2 and C4 components, they form a C5 convertase without need for specific antibody binding representing site. Here we have a small comparison or the chart between the classical, alternative and lectin pathway. Classical pathway is when we have acquired kind of immunity, it activates at that time. But alternative and lectin pathway, when we have an innate immunity, that is a non-specific kind of immunity, at that time they will activate. For the initiation, classical pathway need an antigen antibody complex, while in alternative pathway requires a microbial components, and in lectin pathway it needs a mannose binding or lectin binding site. In classical pathway, the antibodies are needed for the initiation, while in alternative pathway, in lectin pathway, it have a no role. The antibodies does not have any role. The propidine does not play any role in a classical and the lectin pathway, but it plays a very important role for the activation of C3. The C3 convertase, that is one of the major complex, that is C4b and 2b in classical pathway, while in alternative pathway it is a C3b and capital B small b. While the C5 convertase enzyme is same for the classical pathway and the lectin pathway, but it is a different for the alternative pathway. The involved components for the classical pathway are C1 to C9, but in alternative pathway the factor B, factor D and propidine also play a very important role. While in lectin pathway, MBL and MASP1 and MASP2 proteins play a very important role. The MAC formation is formed in all the three stages. Here we have a biological consequences of the complement system. It plays a very important role at the time of cell lysis. It also plays a very important role at the time of inflammation. It plays a very important role for the complement fragments which act as anaphylotoxins. The complement fragments which also act as opsonin, and for the removal of the circulating immune complexes by the complement. So at that time this play a very important role. Here are the references and that's all about it.